You can search for any other database, and you can, for example, a database from NCBI or Genbank or EMBL, you can search for hemoglobin, a human hemoglobin. Like this you can click. It is a second page where you will have different links. And coming to this, okay, of course, as it is a screenshot, it may not be visible, but you go for your internet having this site. After you clicking, you can arrive at this page. Now, BLAST. What is BLAST? It is not a bomb blast. It is an acronym. So, BLAST is the basic local alignment search tool. So, what is BLAST? I repeat, it is a basic local alignment search tool. It is developed during 1990, and in 1997 it is further improvised, and S. Alshel was the main scientist who developed this tool. It is a heuristic method for performing local elements through searches of high scoring segment pairs. So, it is a local alignment. There are different alignments like a pairwise alignment, parallel alignment, and also alignment from different sources. So, BLAST is basically aligning a local segments. That means, first to use statistics to predict significance of initial matches, saves on false reads, offers both sensitivity and speed. So, BLAST can be accessed through NCBI BLAST or Canadian Bioinformatics Resource or European Bioinformatics Institute. Okay. So, I will recommend for NCBI BLAST. This is the NCBI BLAST page. So, you will have the nucleotide, protein, translated, genomes, special and metadata. And what you have to do is you go for a BLAST. Before going to that, different flavors of BLAST, there are different flavors also. What is it? There is a BLAST-P, that is called protein query against protein data bank, and BLAST-N, that is a DNA RNA query against Genbank, BLAST-X 6' trans DNA query against protein DB, and translate, it is a translation nucleotide bank, and T-BLAST-X, it is again 6'. What is 6'? Because a codon will be having 3 bases at one side and 3 bases at one side. It is an open reading frame, it is upward and downward. So, 3 bases and 3 bases, it is called a 6' DNA query, PSA BLAST and also 5 BLAST. So, different formats, I will show you different file formats. It is a Genbank file format. For your understanding, I am just showing only file formats. So, what are the descriptive area in this Genbank file format? Earlier I said that this is a BLAST. BLAT format is different from the Genbank file format. Generally, the Genbank file format is using a particular format called as FASTA. FASTA is developed by Pearson, and here it links a protein. There is a locus, there is a definition, there is a GI name, there is a accession number. Every item you will search from the website. It is available, it is readily available. Okay, it is a Swiss prot. There is a file format as Swiss prot. Now, coming to, due to the lapse of time, I will just show the NCBI BLAST. It is a running NCBI BLAST. It is, you have to give a query. Then, for example, I have given a query on the human protein, and for this sequences, it reads like this. You have to give a FASTA format, raw sequence, or type GI or accession number. Ultimately, you will arrive at this. So, the running BLAST. Choose a range of interest in the sequence set subsequences. Select the database from pull down menu. You will have the pull down menu at the right side. You can search the pull down menu and choose NR for non redundant, and keep his CD search the checkbox on, leave options unchanged, use defaults, go to format menu and adjust number of descriptions and alignments as desired. Okay. I repeat, you have to choose and select NR non-redundant from the dialog box or drop down menu, keep CD search checkbox on, leave options unchanged, use defaults, go to format menu and adjust number of descriptions and alignments as desired. Now, go for select database, a database is selected like NR, the conserved domain database that contains a collection of pre-identified functional structural domains derived from PFAM and SMART databases as well as other sources. Okay. So, this is a next page. When you go for this, you just click on the format after giving the ID number, and when format you will have different format options, and you can be go for a description 100 to 50 alignments. You can go for alignment view pairwise, you can PSA or SciBlast 0.005, and you can select all organisms or any particular organism. You can layout 2 windows and go for blast. Now, it is a result of blast. Now, you will have the blast and it is having with a date when you are going for the blast, the same date will be displayed and reference and query will be answered and your database is this, and it is the output of a blast, it is output of blast. Okay. So, here if you observe the distribution of 43 blast hits on the query sequence, it is complete output and you can go for different varieties of alignments or sequences. Okay. And coming to the ORF, that is up to the blast, the sequencing is done. And what is ORF finder? It is open reading frame finder, it is also a 6 frame. You can type the GI number if you find, if you know, or you can also get down from the drop box. Okay. These are different outputs and see by map over. Okay. As timing is running short, I can show you different formats. You can go through the website using your what you call internet access in your colleges. So, these are different biodiversity databases. Okay. I go to the last slides. I am skipping many issues because of the lapse of time. Okay. Coming to this microarray, you all know the computational drug design, coming to very important bioinformatics application, the computational drug design. For example, usually our doctors prescribe for a patient who is suffering from fever, generally the patient doctor prescribe paracetamol, but you know that the paracetamol tablet, even though it is antipyretic, that means it reduces the temperature, will not work to everyone at that same pace or as efficiently as it suits for someone. That is due to the bioinformatics development or due to the human genome, after the result of the human genome project, there is a tailor-made drug designs. It is the very important, very beauty of the bioinformatics and human genome project. So, you can tailor-made, a paracetamol can be damaged, can cause damage to somebody and can be very good to somebody. So, in future you will be having drug designs based on the human genome project. So, these are genomics and the proteomics and the biopharma, the applications are very enormous, but you have to go for high throughput sequencing, genomics, proteomics, chemoinformatics and combinational chemistry, the molecular modeling, in vitro and in silico models. It is identify disease, isolate protein and also find different drug and preclinical test. After preclinical test only it is administered to human beings. The ligand databases, target protein, molecular docking and ligand docked into protein's active site, everything will be possible only through the bioinformatics advancement. So, there is a knowledge modeling, the study of structure, function and quantity of complex carbohydrates synthesized by an organism. The carbohydrates added to basic protein structure, that is glycosylation. The glycomics a case study. Here you can see the case study of the glycomics. It can be a model, it can be a knowledge modeling, right. So, with all this what is inferred is there is a lot of data coming up every day. This is actually a microarray, it is a chip based, because there are some thousands and millions of dots are there. Each dot will be implanted with molecules, right, and this can be used as chip. Future advancement, that is after the advancement of the Human Genome Project, what is happening is every smart card will be embedded with a chip that is having your DNA. So, in future the health industry will be very much getting benefited from this, because in the smart card what you are having your DNA. So, based on your DNA or genetic mapping, a doctor will prescribe what type of medicine that is useful for you, and it will minimize the risks or minimize the complications that are arised from due to the what is allergic conditions from the different antibiotics or different medicines. For example, if you coming to the SNP, SNPs are also a very important. What is SNP actually? It is a single nucleotide polymorphism, that is a DNA sequence variations that occur when a single nucleotide, different bases like adenine, thymine, cytosine or guanine in the genome sequence is altered. For example, a SNP might change the DNA sequence into different sequence. So, these are very important in curing or diagnosis of cancer also. So, the SNP, that is single nucleotide polymorphs, may have latent effect. Okay. With this I will end up this session, and I advise you to go for internet website and CBI site, and you will be having very good information regarding this, and also search zoology prgc.blogspot.in for different informations. Thank you very much.